Welcome to the class, everyone. We are starting today germplasm conservation and cryopreservation. As we can define the germplasm as hereditary material, that is total content of genes transmitted to the offspring through germ cells. Germplasm provides the raw material for the breeder to develop various crops. Thus, conservation of germplasm assumes significance in all breeding programs. Germplasm is necessary for the conservation of all the genes which are present in any given crop species, and if we have collected all the germplasm of any said species, then we can do breeding program for the improvement of particular crop species for higher yield, higher productivity, and for the protection against various biotic and abiotic stresses. As the primitive man learned about the utility of plants for food and shelter, he cultivated the habit of saving selected seeds or vegetative propagules from one season to the next one. Because the plants can be propagated by two means, either by seed or by vegetative propagules. So this is traditional way of propagating plants, saving seeds or propagules from one season, and then these are used in the next sowing season. In other words, this may be regarded as primitive, but conventional germplasm preservation and management comes from this, and it is highly valuable in breeding program, as if we are going for the locally cultivated traditional crop varieties, then we are conserving the germplasm in broad ways, and it is highly valuable in crop improvement programs. The objective in germplasm conservation mainly is to preserve the genetic diversity of a particular plant or genetic stock, and then it can be used for any time in future in the crop improvement program. But in recent years, many new plant species with desired and improved characteristics have started replacing the primitive and conventionally used agricultural plants. It is important to conserve the endangered plants, or else some of the valuable genetic traits which are present in the primitive plants may be lost, and that will be loss of biodiversity of particular crop species. A global body, namely International Board of Plant Genetic Resources (IBPGR), has been established for germplasm conservation. The main objective is to provide necessary support for collection, conservation, and utilization of plant genetic resources throughout the world. In India, germplasm collection is undertaken by National Bureau of Plant Genetic Resources. Headquarters are situated at Delhi. There are two approaches for germplasm conservation of plant genetic material: in situ conservation and ex situ conservation. If we talk about in situ conservation, it means the conservation of germplasm in their natural environment, in the original habitat, by establishing biosphere reserves or national parks or gene sanctuaries. Then it is termed as in situ conservation. This approach is particularly useful for preservation of land plants in a near natural habitat, along with several wild relatives with genetic diversity. In situ conservation is considered as a high priority germplasm preservation program because here plants are being conserved in their natural habitat. The major limitations of in situ conservation are the risk of losing germplasm due to environmental hazards. If sometimes there are flood or any other natural hazards, then we can lose these naturally growing germplasm spots. The cost of maintenance of a large number of genotypes is very high as we have to maintain them in living conditions in fields. So field is required and the labor is also required. Ex situ conservation is the chief method for the preservation of germplasm obtained from cultivated and wild plant material. Genetic material in the form of seeds or in the form of in vitro cultures like plant cells, tissues, or organs can be preserved as gene banks for long-term storage under suitable conditions. For successful establishment of gene banks, adequate knowledge of genetic structure of plant populations and the techniques involved in sampling, regeneration, maintenance of gene pool, etc. are essential. In case of plants, ex situ conservation also can be carried out by establishing botanical parks or botanical gardens. Germplasm conservation in the form of seeds, the easiest method. Because seeds are the most common and convenient materials to conserve plant germplasm. This is because many plants are propagated through seeds and seeds occupy relatively very small space. Seeds can be easily transported to various places. For this, you have to solar dry the seeds, then you can treat them with some chemicals like fungicide, etc., and then you can store them for a longer duration at low temperature. But there are also certain limitations in seed conservation, like viability of seeds may be reduced or lost with passage of time. Seeds are susceptible to insect or pathogen attack, often leading to their destruction. This approach is exclusively confined to seed propagating plants and therefore it is of no use for vegetatively propagated plants like potato, etc. And it is difficult to maintain clones through seed conservation. Certain seeds are heterogeneous and therefore are not suitable for true genotype maintenance. In vitro methods for germplasm conservation employing shoots, meristems, and embryos are ideally suited for the conservation of germplasm of vegetatively propagated plants. Plants with recalcitrant seeds and genetically engineered materials can also be preserved by this in vitro approach. There are several advantages which are associated with in vitro germplasm conservation. First, large quantities of materials can be preserved in small space. Germplasm preserved can be maintained in an environment free from pathogens, that is aseptic environment. So there won't be establishment of any type of disease on the germplasm which have already gone for germplasm conservation. It can be protected against the nature's hazards when you are protecting your germplasm in vitro condition, in lab conditions, or in some seed vault, then you are protecting your material from nature's hazards also. From the germplasm stock, large number of plants can be obtained whenever needed by multiplying the material which has undergone the conservation. Obstacles for their transport to national and international borders are minimal since the germplasm is maintained under aseptic conditions. These are three approaches for in vitro conservation of germplasm. First is cryopreservation, then there is cold storage, and then there is low pressure and low oxygen storage. Cryopreservation, we will discuss this in detail as cryopreservation is the most preferred method for long-term germplasm conservation. Cryopreservation has been taken from a Greek word cryos that means frost or in frozen condition. It means preservation in the frozen state. The principle involved in cryopreservation is to bring the plant cell and tissue cultures to a zero metabolism or non-dividing state. That means at such state where there is no cell division in the plant cell and there is no metabolic activities going on. How it can be done? It can be done by reducing the temperature in the presence of cryoprotectant. So that cryoprotectant gives the protection to the plant cell and there is no damage to the plant cell at such low temperatures, even ultra low temperatures. Cryopreservation broadly means the storage of germplasm at very low temperature. It can be achieved first over solid carbon dioxide at minus 79 degrees centigrade. At low temperature deep freezers around minus 80 degrees centigrade. In vapor phase nitrogen minus 150 degrees centigrade and in liquid nitrogen at minus 196 degrees centigrade. Among these, the most commonly used cryopreservation is by using liquid nitrogen. At the temperature of liquid nitrogen, that is minus 196 degrees centigrade, the cells stay in a completely inactive state and thus can be conserved for long periods. In fact, cryopreservation has been successfully applied for germplasm conservation for a wide range of plant species, for example, rice, wheat, peanut, cassava, sugarcane, strawberry, coconut. Several plants can be regenerated from cells, meristans, and embryos which are stored in cryopreservation. What is the mechanism of cryopreservation? The technique of freeze preservation is based on the transfer of water present in the cell from a liquid to a solid state. Due to the presence of salts and organic molecules in the cells, the cell water remains in liquid state and it requires much more lower temperature to freeze even up to minus 68 degrees centigrade according to several reports compared to the freezing point of pure water that is around 0 degrees centigrade. When stored at low temperature, the metabolic processes and biological deteriorations in the cells or tissues almost come to a standstill. Precautions or limitations for successful cryopreservation. First, you require good technical and theoretical knowledge of living plant cells and as well as cryopreservation techniques. Other precautions for successful cryopreservation are: first, formation of ice crystals inside the cell should be prevented as they cause injury to the organelles and the cells. This is the most important task to be undertaken when you are going for cryopreservation of plant cells. Second, high intracellular concentration of solutes because it may also damage the cells. This concentration of solutes may be toxic or damaging to the plants. Sometimes certain solutes from the cell may leak out during freezing. Cryoprotectants also affect the viability of cells and the physiological state of the plant material is also important. Technique of cryopreservation. This is an outline of the protocol for cryopreservation of shoot tip that has been depicted in the figure shown on next slide. The cryopreservation of plant cell culture followed by the regeneration of the plant broadly involves these following stages. That is the whole technique of cryopreservation. First is development of a sterile tissue culture. Then second, addition of cryoprotectants or pre-treatment with cryoprotectants. Number 3, freezing. Number 4, storage. After that, thawing, reculture, measurement of survival or viability, and lastly, plant regeneration. So, in this picture, the shoot tip culture. These are the shoot tips which have been pre-grown on medium with DMSO. DMSO is dimethyl sulfoxide, that is a widely used cryoprotectant. Here, the shoot tips which are present in semi-solid medium They are plunged into medium at 25 degrees centigrade, then shoot tip plunged into liquid nitrogen, and there is the shoot tips in ampule which are frozen in liquid nitrogen. They are stored in liquid nitrogen, and whenever required, this ampule is taken out, thawing is done, and then we have these shoot tips on semi-solid medium. So this is the first step, that is development of sterile tissue culture. You have studied it already. The selection of plant species and the tissues with particular reference to the morphological and physiological characters largely influence the ability of the plant to survive and cryopreservation. Any tissue from a plant can be used for cryopreservation, for example, meristem, embryo, endosperm, ovule, seed or cultured plant cells, protoplast, callus, etc. Among these, meristematic cells and suspension cell cultures in the late lag phase or log phase are most suitable. Second step, addition of cryoprotectant. Cryoprotectants are the compounds that can prevent the damage caused to cells by freezing or thawing. Freezing point and supercoiling point of water are reduced by the presence of cryoprotectant. As a result, the ice crystal formation is retarded during the process of cryopreservation. There are several cryoprotectants which include DMSO, dimethyl sulfoxide, glycerol, ethylene, propylene, sucrose, mannose, glucose, proline, and acetamide. Among these, DMSO, sucrose, and glycerol are most widely used. Generally, a mixture of cryoprotectants instead of a single one is used for more effective cryopreservation without damage to cells or tissues. Next step is freezing. The sensitivity of the cells to low temperature is variable and largely depends on the plant species. There are four different types of freezing methods. First is slow freezing method. The tissue or the requisite plant material is slowly frozen at a slow cooling rate of 0.5 to 5 degrees centigrade per minute from 0 degree to minus 100 degrees centigrade, and then transferred to liquid nitrogen. The advantage of slow freezing method is that some amount of water flows from the cells to the outside. So this will promote extracellular ice formation rather than intracellular freezing. As a result of this, the plant cells are partially dehydrated and survive better. A slow freezing procedure is successfully used for the cryopreservation of suspension cultures. Second method is rapid freezing method. This technique is quite simple and involves plunging of the vial containing plant material into liquid nitrogen. During rapid freezing, a decrease in temperature minus 300 degree to minus 1000 degrees centigrade per minute occurs. The freezing process is carried out so quickly that small ice crystals are formed within the cells, and the growth of intracellular ice crystal is also minimal. Rapid freezing technique is used for the cryopreservation of shoot and somatic embryos. Number three, stepwise freezing method. This is a combination of slow and rapid freezing procedures with the advantage of both and is carried out in a stepwise manner. The plant material is first cooled to an intermediate temperature and maintained there for about 30 minutes and then rapidly cooled by plunging it into liquid nitrogen. Stepwise freezing method has been successfully used for cryopreservation of suspension cultures, shoot apices, and birds. Last one is dry freezing method. Some workers have reported that the non-germinated dry seeds can survive freezing at very low temperature in contrast to water imbibing seeds, which are susceptible to cryogenic injuries. In a similar fashion, dehydrated cells are found to have a better survival rate after cryopreservation. Next step is storage. Maintenance of the frozen cultures at the specific temperature is as important as freezing. In general, the frozen cells or tissues are kept for storage at temperatures in the range of -70 to -196 degrees centigrade. However, with temperatures above -130 degrees centigrade, ice crystal growth may occur inside the cells, which reduce viability of cells. Storage is ideally done in liquid nitrogen refrigerator at -150 degrees centigrade in the vapor phase or at -196 degrees centigrade in the liquid phase. The ultimate objective of storage is to stop all the cellular metabolic activities and maintain their viability. For long-term storage, temperature at -196 degrees centigrade in liquid nitrogen is ideal. A regular and constant supply of liquid nitrogen to the liquid nitrogen refrigerator is essential. It is necessary to check the viability of the germplasm periodically in some samples. Proper documentation of the germplasm storage has to be done here. The documented information must be comprehensive with the following particulars. First, taxonomic classification of the material. Second, history of culture. Third, morphogenic potential. Fourth, genetic manipulations done. Fifth, somaclonal variations. Then culture medium, and last one is growth. Thawing is usually carried out by plunging the frozen samples in ampoules into a warm water. Temperature is around 37 to 45 degrees centigrade of water bath and with vigorous swirling. By this approach, rapid thawing occurs and this protects the cell from the damaging effects of ice crystal formation. As the thawing occurs up to the point when ice starts melting, the ampoules are quickly transferred to a water bath at temperature 20 to 25 degrees Celsius. This translates for long time in warm water bath. For the cryopreserved material, cells or tissues, when the water content has been reduced to an optimal level before freezing, the process of thawing becomes less critical. Reculture. In general, thawed germplasm is washed. The material is then recultured in fresh medium following standard procedures according to the protocol which has been already optimized. Measurement of survival or viability, it can be done with the help of several staining techniques using triphenyl tetrazolium fluoride (TTC), Evans blue, and fluorescein dye-ST. These are commonly used staining techniques and the best indicator to measure the viability of cryopreserved cells is their entry into cell division and regrowth in culture. And this can be evaluated by the following formula: number of cells or organs growing divided by number of cells or organs thawed into 100. Plant regeneration. This is the ultimate purpose of cryopreservation, to regenerate the desired plant. For appropriate plant growth and regeneration, the cryopreserved cells or tissues have to be carefully nursed and grow. Addition of certain growth promoting substances besides maintenance of appropriate environmental conditions is often necessary for successful plant regeneration. A selected list of plants have been successfully used for cryopreservation that has been shown on the next table. In cell suspension, Oryza sativa, that is rice, glycine max, soybean, zea maize, Nicotiana tabacum, and capsicum annum has been preserved. In the form of callus, Oryza sativa, capsicum annum, saccharum species has been preserved. In form of protoplast, it has been used for cryopreservation of zea maize and Nicotiana tabacum. Meristem has been used in Solanum tuberosum and Cicer aratinum. Zygotic embryos has been used in zea maize, Podium vulgare, and Manihot esculenta. Somatic embryos have been used in citrus sinensis, Daucus carota, and Coffea arabica. Pollen embryos have been used in Nicotiana, citrus, and Atropa species. Applications of germplasm storage are immense for plant breeders and biotechnologies. They are used as maintenance of stock cultures. Plant material of several species can be cryopreserved and maintained for several years, and it is used as and when needed. This is in contrast to an in vitro cell line maintenance, which has to be subcultured. Here, there is no need of subculturing. So, the germplasm storage is an ideal method to avoid subculturing and maintain cells or tissues in a viable state for many years. Cryopreservation is an ideal method for long-term conservation of cell cultures which produce secondary metabolites which are used in various sectors by humans. Disease-free plant material can be frozen and propagated whenever required. Recalcitrant seeds can be maintained for long durations. Conservation of somaclonal and gametoclonal variations in cultures can be done. Plant materials from endangered species can be conserved. Conservation of pollen for enhancing longevity. Rare germplasm developed through somatic hybridization and other genetic manipulations can be stored. It is a good method for the selection of code-resistant mutant cell lines which could develop into frost-resistance plants and establishment of germplasm banks for exchange of information at the international level. There is limitation of germplasm. Major is it is the expensive equipment and trained personnel which are required for germplasm conservation in vitro. For the cryopreservation, it may, however, be possible in the near future to develop low-cost technology for cryopreservation of plant material, and it will benefit us in long way. Thank you so much for listening carefully. Thank you.